So this is a sort of a new area for me in work. I've worked with wastewater treatment plants since I was a graduate student in water treatment plants and various aspects of them. And on some occasions, I've worked with the folks in EHS. I've enjoyed that very much, particularly with the ES&E students and professors Suffet, Ambrose, and Weiner. We'll move forward with our presentation. So the introduction, and I always try to provide a slide in my presentations that talks about where we're going. And so I'm going to say something about antibiotics and then ARGs and ARBs and why is it a problem and why I think we can have some solutions. So this is a table from the World Health Organization that talks about pathogens and the potential for antibiotic resistance. And they've characterized them as critical, high, and medium. And this is a medical field, so I'm not producing this, I'm just responding to it. But you can see these pathogens that are potential problems. And antibiotic resistance is a big concern, not only among us engineers, but especially the medical community. And some of us have been lobbying government to try to support more antibiotic development. And it's frustrating to know that developing a new drug costs about $2 billion in several years. And so unless there's a great potential for use of this new drug, private industry is not likely to develop it. And so that's the problem with vaccines, although the COVID vaccine is getting a lot of attention. But vaccines are used once or twice every few years, and drug companies would rather develop something that you're going to take every day. So we need to worry about these sorts of diseases, and the worst of the predictions are that we're going back in time to a period before antibiotics, when people died of diseases that we've really wiped out now. And it comes close to home too, because I can think about my daughter. When she was about 3 years old, she was getting ear infections, and her pediatrician looked at her ear and said, well, I see there's an infection. We'll try amoxicillin, but I'm pretty sure it's not going to work because of antibiotic resistance. And that was true. It had no effect whatsoever. And then we tried Augmentin, which was another drug, which was also resistant. And the third drug was a new one, which did work. And so this antibiotic resistance is already close to home here, and we need to be concerned about it. So this is a slide from a presentation that Renjay developed for the Water Environment Federation, which we presented this week. The Federation is a large meeting, and it was done online this year in quite a different fashion. But what can happen is that we get gene transfer in a treatment plant from one bacteria to another bacteria. And there are different ways of having gene transfer. I'm not going to spend much time on it, but basically it means that one bacteria that's resistant to an antibiotic can transfer a gene to another bacteria that might have very different survival characteristics or places in the environment. So we worry about that change. And so I want to talk now about wastewater treatment plants. And this is a picture also from Renjay's seminar, but you can see the reference to it down to the left. And we see they have a sort of central spot in this arena where we have livestock and aquaculture, things we eat that go into the community, and hospitals, pharmaceutical industry, drinking water, and they're discharged to the environment. Some come directly discharged, and some will go into aquifers and into soil. So the treatment plants there are in the middle, and they take waste from different places, and treat it, and then we hope it's discharged into the environment. So if the treatment plants create antibiotic resistant genes from different inputs, because you see there are hospital inputs, pharmaceutical industry inputs, lots of other industries, then they can be discharged to the environment and perhaps come back to haunt us. So we need to understand how wastewater treatment plants work. And I'm going to allude to a paper we wrote that was a literature review of all the work that was done with treatment plants and antibiotic resistance. And we're really in a very diverse field, multidisciplinary, because the folks doing molecular biology to detect genes are usually not associated at all with treatment plants. And treatment plant engineers rarely know anything about molecular biology. So there's two groups with a large gulf between them. So treatment plants have been constructed over the past 100 years or so. Their original purpose was to protect human health, either by segregating the waste from drinking water or contact with humans. So the original treatment plants were little more than sewers that just conducted the waste away from folks. And if you're a history buff, you can learn about Rome and its success as a 500-year-old city was largely associated with the way it handled its waters and wastewaters. So later, these treatment plants became much more important for environmental protecting, preventing fish kills and other negative impacts from both municipal and industrial waste. And so we had landmark legislation in 1972. Richard Nixon was president. It was a great time for the environment. It was the first Earth Day. There were lots of things going on. And we had the amendments to the Clean Water Act, PL 92-500. And that's significant because it required all discharges to receive at least secondary treatment. And secondary treatment at the time was defined as 85% removal of BOD and TSS. And at the time, it was a good definition because it really squelched all the arguing that could be done over what was important. But it's no longer a complete definition because BOD and TSS says nothing about a lot of toxic materials and even antibiotic resistant genes. So we don't know exactly what our treatment plants are going to do. So we've developed a lot of better and more advanced treatment plants to remove pathogens, nutrients, and toxic materials, and aesthetic impacts, color and odor. But we haven't been too concerned about antibiotic resistance removals. And so if we go back and look at treatment plants, we can understand how they work. The early examples, mostly in England, worked by capturing a natural assimilative process that occurred in streams and lakes and putting those processes into an engineered process. So we observed the pollution in the river, and it grew voluminous quantities of bacteria and caused all these water quality problems. And somebody said, well, why can't we confine that growth to a treatment system and protect the river? And so let's grow them in a process segregated from the environment. And the key to making these processes work were to provide sufficient oxygen and a media that could contain the bacteria. And so several processes involved to treat the waste. These were treatment techniques, trickling filters, lagoons, and finely activated sludge plants. So we and other industrialized nations have been building and operating treatment plants of different sorts, but almost all use bacteria or protozoans to metabolize the pollutants. So here are 4 pictures that I just included because I know many of you aren't familiar with treatment plants. But the upper left here is a trickling filter plant. And trickling filters are an early technology, and they simulate what happens when a river flows through a rocky rapids. And you can see the arm spreads the wastewater out among this media. And these were employed in England very early on, and they use coal for media. On the right, we see a lagoon. Lagoons are sort of a lake dedicated to wastewater treatment. And the The particular one you're looking at here is north of Mexico City. It's one of the giant stinking lagoons that treats the waste of Mexico City. And I took this picture when I was on a trip to Mexico as part of a joint project with EHS. And Professor Suffet was standing behind me when I took this picture. So on the lower left, we see Hyperion, which is a plant that was constructed finishing about 1998. And it's what we call a high purity oxygen plant. And you can see the white surface there with the little purple dots. Those are aerators, and the orange things in the background are clarifiers. And then on the extreme right is something new. It's a gray water treatment system with plants. And this is a research project that we had in engineering to treat gray water from houses for reclamation. And that was, the gray water excludes toilets and sinks from kitchens and so forth. It's mostly treating the showers and the laundry waste. But what's common about all four of these processes is that they use this bacteria to metabolize the waste. We put them to work for us. And it's really good. They work hard, and they're smart, and they don't have union cards. So we're really fortunate to be able to metabolize these wastes. So what can happen, though, is we get gene transfer during this process. And the environment in these treatment plants is an excellent environment for bacterial growth and gene transfer. And we've actually depend upon gene transfer to make them work. Because we say activated sludge, it's called activated because the bacterial community evolves and is enriched to remove specific compounds by metabolizing them. So when we start up a biological process, and it receives a new compound, such as an industry that starts making some new product that has a particular waste. At first, the biological process may not remove it, and it may be toxic to the biological process. But after a short period, the organisms acclimate to remove it well. And this is the reason why there's so much emphasis on biodegradability of products such as cleaners, paints, and solvents, and so forth. We want them all to be biodegradable. And biodegradable is a relative term. There are a few things that are just absolutely not biodegradable, but there are many things that biodegrade so slowly that we can't biodegrade them in our treatment plants. So our plants will acquire the ability to metabolize these compounds and protect the environment. And I'm reminded of a movie where Jeff Goldblum said, nature always finds a way. That may not be quite correct, but I think it's 99% correct. So nature finds a way. So the most successful of these processes keep the bacteria as long as possible in order to transfer and keep slow growing bacteria that can metabolize complex compounds. This environment also provides a good environment for bacteria to become acclimated to antibiotics. If we discharge antibiotics into our treatment plant, we give those organisms an opportunity to acclimate to the antibiotics to remove it. And so that's why we're worried about antibiotic creation at treatment plants. And that's why the treatment agencies have all developed programs to encourage people to not flush antibiotics down the toilet, to dispose of them in proper ways. So I've been concerned about this, and a lot of my colleagues in the environmental engineering field. And so we wanted to do research in this area. And I was convinced that the research going on was performed by two disparate groups who really didn't communicate with each other. And so a good way to get started in any field, especially if it's a new field, is to perform a literature review. And so this is a paper we published, it's almost 2 years ago. Renjay is the first author. He worked a year on this paper, collecting all the references. And Ginny Jay was Renjay's master's advisor and has helped us with equipment and doing the analytical techniques. So it was published in Water Environment Research, and I'm pleased to report that we won a prize for it that was just announced this week. It was considered the best publication in the Water Environment Federation for that year. And so what we did is we reviewed all the published literature on ARGs and ARBs at treatment plants, antibiotic resistant genes and antibiotic resistant bacteria. And we found that most or many of the researchers doing molecular biology did not understand treatment plants. I contacted some of them because I wanted information on what they were studying. And believe it or not, I got a couple of comments. You mean there is more than one type of treatment plant? So it was challenging to understand their results in some cases, and we contacted these authors. Sometimes they would talk to us and sometimes they wouldn't. So in our review, we found a lot of information on these particular antibiotic resistant genes. They're listed here towards the drug that they're resistant to, tetracycline. Tetracycline is one of the most important antibiotics. It's been with us a long, long time. And it's used also on farm animals. It's probably used more on farm animals than on humans. And so we see the different genes as we go down. And of course there are more, but this is what we were able to find in the literature. And so what we see here are some of the results of this survey. And so at the bottom you see the types of processes. ASP, activated sludge process. That is the most common wastewater treatment technique in the United States. It's more high-tech than other things, but the Hyperion plant is an activated sludge process, and there's about 15 activated sludge plants treating all the wastewaters in the city of Los Angeles and Los Angeles County. And it tends to be more expensive and more complicated. And then along the rest of the horizontal axis, you see UV and anaerobic digestion and so forth. There were other processes that were commented upon. Many of these exist at treatment plants. And so the two colors that you see were the results that we observed. And it's just listed as an increase or a decrease. The dark blue is an increase. And so we have about 75 observations for activated sludge plants, and maybe about 65 of them showed a decrease in the absolute concentrations of antibiotic resistant genes and bacteria, and about 10 of them showed an increase. And as we go down, we can look at some other things. And what we want are all of these processes to kill off all the antibiotic resistance and prevent it from entering the environment. So this is another picture of the results, but there's more detail here. And again, we see the horizontal axis, which is the type of process, and activated sludge is on the left. And I put MBR there because folks tend to think that MBR is something different, but it's just a type of activated sludge process. And then on this slide, you can see the different genes that were reported. And the light blues are tetracycline. And you can go across and see what the resistance and the survivability was. And by the way, I can make the paper available, which has a lot more detail. And so this is another summary picture that's a little bit a repeat of the previous information, but it's displayed a little bit differently. And there you see activated sludge, UV, anaerobic digestion, and so forth. And here we're listing observations unchanged, observations increased, and observations decreased. So the net result of our survey was that for the most part, we found a decrease in antibiotic resistance through the treatment plant. And most of these studies were looking at the primary effluent, which enters the activated sludge process or trickling filter, and the final effluent before disinfection. You can see UV and ozone and so forth. Those are disinfections. And they are effective in reducing bacteria introduced to the environment, but it wasn't the focus of our research. And so what we wanted to do was to further these results and to study the fate of these genes in treatment plants. And I'm a treatment plant expert. I'm not a molecular biologist, but I know of ways to optimize treatment plants. And we've done that and published that on ways to optimize treatment plants to remove refractory organics, things like drugs and so forth. The easiest example to cite is ibuprofen, which is Advil. At a certain type of treatment plant, it goes through untreated, almost unchanged. And then you can operate the plant a little differently, maybe 5% more expensively, and you can affect removal down to detection limits. So I was most curious about how we could operate the treatment plant to optimize these removals. And so that's to study the relationship between the fate of ARGs and ASP. And the third thing I wanted to do was to provide a feasible introduction of the impact of SRT to the fate of ARGs. SRT is the solids retention time. It is the key parameter in running a treatment plant, and it affects what sorts of things you can remove. And if I go back to the Clean Water Act, the 85% removal can be achieved at low SRT or high SRT. And I indicated that TSS and BOD weren't sufficient to monitor all of these other things we want to remove. And so we're really trying to optimize the treatment plants with some new parameters and new operating strategies. And the old guard of the treatment industry is reluctant to change some of these practices. So our methodology, sample collection, 4 treatment plants, 8 weeks, 1 sample per week. There's a lot of variability in the analysis, and we decided to do 8 samples and average the results over 8 weeks. And we did a DNA extraction and then a qPCR in Professor Jay's lab, and then we did statistical analysis of the results. So those are the genes we were looking at: SOL1 and 2, Tetra W, and 16-RSNA. And here's some of the calculations we did with the statistical analysis, in addition to things like standard deviations and analysis of variance. And I'll point out one thing on this slide here: VSS, volatile suspended solids. That is a parameter that is used in treatment plant operation. You probably won't find it in a real chemistry lab at all. But what it is, it's the residue of a filtration through a 0.5 micron filter. And it's the volatile part of it. And volatile in this case means organic. So the analysis excludes precipitates like calcium carbonate and other metals, and is only the organic part. So in operation of treatment plants, we find VSS very useful in controlling the plant and understanding its efficiency. So one of the things I wanted to do was to see if we could adjust our treatment plant operation on antibiotic resistance based upon something that treatment plant operators know and understand, which is VSS, or volatile suspended solids. So here are the treatment plants, and I don't put their names down, although you could probably figure them out. And they're here in Los Angeles, within an hour's drive of UCLA. And Renjay was the person that went out and took the samples. And so you can see the 2 types there: long SRT, MLE ASP, short SRT, and HPO ASP. Now, those operating techniques I don't think were known to any of the molecular biologists, but they're really important if you're a treatment plant person. So the high purity oxygen ASP is a type of plant that's used most often in big cities. Los Angeles has one, Orange County has one, San Francisco has 2. Seattle has one, East Bay Mud has one, Detroit has the largest one, and I could go on and on and on. But they're short SRT plants, 1.5 to 2 days. Now what the SRT means is that's the average or mean life of a microorganism that's in the process. So if you're going to run a treatment plant at 2 days or less, whatever bacteria are going to be retained in that process have to multiply in less than 2 days. So that means that you select against slow-growing bacteria. And that shows up in removal efficiencies, because these short SRT plants cannot remove complex organic compounds that take longer to degrade. So the top 2 plants are longer SRT plants, and they are capable of removing nutrients. And one of the reasons we go to them is to remove nitrogen and phosphorus, which are both important nutrients to remove in order to prevent eutrophication of receiving waters. So the 2 parameters on the right, or the 3 parameters on the right, are indicators of the process. The primary effluent, I'll show you a picture in a second, TSS, the average secondary effluent, TSS, and TSS is total suspended solids, and it includes the non-volatile part. So it's the VSS put any minerals like calcium carbonate. And usually the VSS is about 80% of the TSS, so it's not too much different. It's a little easier to measure TSS. And so you see the primary effluent has high numbers, which is to be expected. Primary treatment only removes about 35% of the pollutants. And the secondary effluent are 2 sets of numbers here. And you can see 5 and 3 and 17 and 13. The 5 and 3 represent excellent TSS removal, and this is without filtration, and it's achievable in these long SRT plants. And over the past 10 years, I've published lots of papers urging treatment plant agencies to adopt longer SRT operation, even when they don't have to nitrify. And then the other 2 plants are short SRT, and you invariably have higher TSS removal and higher TSS concentrations. And I want to plant the seed that if the antibiotic resistant bacteria and genes are released from the treatment plant through these total suspended solids, and that's what we're measuring, then the short SRT plants would be worse because they're just going to remove more solids. And then on the right, you see the flow rates of the treatment plants. Los Angeles has 2 of the very largest treatment plants west of the Mississippi. And so this is a process flow, and I'll just go through 1 or 2 of them here. But on the left, you see pre-treatment, bar screens and stuff. That removes junk that gets into sewers. And then you have primary treatment, which is settling. And prior to 1972, the Clean Water Act amendments I mentioned earlier, most large cities only had primary treatment. And if I take you out to a treatment plant and I show you primary effluent, the result of primary treatment, you'll say, yuck, it'll have an odor, and you'll say, how can we release that to the environment? But that was standard procedure before 1972. And then secondary treatment is usually a biological treatment. It grows these organisms that I've been talking about that are acclimated, and we can run the process to retain slow-growing bacteria, or we can run the process to retain only fast-growing bacteria. And then you have a secondary clarifier, which separates the solids. And then you see the post-treatment techniques, filtration, chlorination, and perhaps reclamation of the effluent, which is becoming very common now. But where we took the samples were downstream of the secondary clarifier and downstream of primary treatment. So we didn't want to complicate the results with the effects of disinfection. So here we see some of the results of our work. And the vertical axis is a gene copies per milliliter. So the higher the number, the more antibiotic resistance is being measured. And across the horizontal axis are the plants, 1, 2, 3, and 4. And then the different color bars are the different genes that we found. And so the crosshatch ones are effluents, and the solid ones are influence. So you can see, if we look at plant 1, that the SOL1 gene was approximately about 8.8 logs coming in. And you can see the whiskers there that define the variability of our measurements. And then the release is next to it in light blue, crosshatched. And we're down to about 8.8. So we had about 2 logs removal, which would be 99% removal. So that was good news. And then if we look at the others across the page, we can see better removals of these genes. There is some differences in the efficiency for the different genes, but in general, the plants were successful at removing them. So this is the absolute gene log removal efficiencies. And I mentioned 2 logs removal, or 1 log removal from the previous picture. So you can see what sorts of removals we got. And this is average for all 4 plants for different weeks. And you can see the variability and why it was necessary to collect multiple samples. But there's quite a bit of variability, because we've got this on a logarithmic axis, so one unit is a factor of 10. And so this is SOL2. This was SOL1 here, SOL2, which was lower in concentrations, but had pretty good removal. And now we look at tetracycline, which I believe was the worst performing. And that might be unfortunate, because it's one of the ones most used. And you can see some negative results here, which we think is due to an interference, because wastewaters, particularly large cities with industries discharging, can have interferences that make your life complicated when you do analyses. And so here's relative abundance. And 2 things are important. One is the absolute number of these antibiotic-resistant bacteria that you have. And the other is the approximate frequency of antibiotic resistance among them. So you don't want 100% of the bacteria to be antibiotic resistant in your effluent. And so we begin to see some increase in the relative abundance. And this graph is divided up into 2 parts. And the top part is the log gene copies per milligram of VSS. That's the new thing I told you about. And the lower part is the log genes copies of 16S RNA. And so the molecular biologists are going to measure 16S, and that's going to be proportional to the total bacterial mass that's present. And for treatment plant people, the VSS is important. So what I wanted to show with this was that we could relate these relative abundances to the VSS. Because if we can tell treatment plant people to reduce the VSS in their discharges, they'll know what to do and they'll have solutions. If we tell them to reduce the 16S RNA, they'll say, what's that? And so this is the correlation that shows the 4 plants. And we're continuing this work, as you'll see in another slide. And so these correlations are sort of preliminary. As we get more data, we will include more information. But on the horizontal axis, you can see the log of the milligrams of VSS per milliliter. That's a concentration. And on the vertical axis, we see the gene copies. And so you can see that there is a positive correlation. So we have a chance of giving treatment plant operators a parameter that will help them with their antibiotic resistance. So this almost concludes the presentation. The complete analysis of the current 4 plans we've completed it, but it's not part of this demonstration. We've added more replicate analysis to reduce the variability. Our work is going to continue. We're going to pick up another treatment plant or two, and we also said in our proposal that we would try to understand the fate of heavy metals on ARGs. Heavy metals in a treatment plant are best to be avoided, but we get them nevertheless. And they can change the microbial ecology in the treatment plant because the organisms are going to try to acclimate to the presence of metals. And some metals are quite toxic, and some can be managed by the bacteria. For example, mercury entering a treatment plant, it's hard for the bacteria to manage that. But chrome can be converted to trivalent chrome, and you can have actually large concentrations of chrome in the mixed liquor that is tied up in the biomass. And so we want to develop a methodology to translate the results so that treatment plant operators and designers can appreciate it. So thank you for participation. These are the authors, and like I say, Renjay's in the audience, and he may help me with questions. So let's see, I've forgotten what time it is here. It's 4:44, so that's about the right amount of time.